All right, so now what we're going to do is look at this topic called gene expression. Right? And gene expression really encompasses two separate processes, transcription and translation, which people often confuse because they have similar sound to them. The idea is that we talked about DNA a bunch, and DNA replication and cell division, which is why the DNA replicates. That's one aspect. So the DNA codes for information within the cell. It has to be replicated in order for the cell to divide, and that's one process. But that's not the reason DNA exists. The reason DNA exists is to code for information, and that whole replication process has nothing to do with the information coding or the decoding of the information. That's what these processes have to do with. So when we look at a single chromosome and all the double-stranded DNA, what you'll find is that it's broken up into regions. And there could be many of these regions. And each of these regions would correspond to a single gene, right? So that would be a gene, that might be a gene, this is a gene here, and so on. So first off, what is a gene? The gene is a sequence of DNA and it codes for a functional product. Okay, so first off, it's going to be within the DNA we're talking about. We're talking about a region of DNA here. And the best definition here is going to be codes for a functional product. And the reason I'm giving you that definition as opposed to one you might look up, and it might say genes code for proteins. That's very old understanding of how this sort of works. Genes code for pieces of information that is going to be directly used to make RNA. Right, so transcription is going to be our process of going from DNA into RNA. So that's technically what do genes code for? They code for pieces of RNA. That is specifically what they do. Right. Now, is that the end point? No, that's not. They're not just to make the RNA. The purpose. So, what's the purpose of RNA? The purpose of RNA is to then make polypeptides or proteins. So, in the next process that we get into, translation, is where RNA will be used. Not that simple, but basically to make what we call polypeptides. Also, and under certain conditions, referred to as a proteins. The main difference here is that the protein has a function. The polypeptide is structural, and generally they can have the same overall structure, but if some part of the structure is missing or not folded correctly, it has no function. If there's no function, it's polypeptide. If it has one, then it's the protein. So it's kind of like very slight definitions, but it's difference, but there is a difference. So the genes are within the DNA, and what we have to do is first copy them. Well, the thing is about that is we don't necessarily copy them all at the same time. There are many genes in your chromosomes that are never decoded or read. The information is just there. They're sort of ancestral genes that might code for things, and we don't use them. And so they're never, ever expressed at all, or in any case. Right, so not every gene is expressed. Not every gene is expressed all the time. Some genes are being expressed at certain points in time, and then they can be stopped. Right? They're repressible. They can be held back. Right? Others are off most of the time. They're not on. They're not, but they could be, and sometimes under certain circumstances, they are turned on. So they're called inducible. They can be turned on, so switched on or switched off. And then there's some things that we need all the time. We always have to have certain proteins in the cell, certain enzymes to carry out certain jobs, and cells are always using those genes. They're getting information from those genes, and they're making functional proteins from the information within them. So that's kind of like a big picture, idea. So what we're going to focus on here first is the process of transcription. And so again, big picture, we're not going to be focused on the whole entire chromosome. We're really just looking at a piece of the chromosome. And what's going to happen is the DNA all right, there we go. Looks a little bit like DNA replication, right? So these two strands of DNA are going to be pulled apart, right, in this region here. Not both of the strands are going to be used for information. What's going to happen is there's going to be, first off, the identification of where to start. So where does a gene begin? Right? So we'll have a region of DNA, called a promoter. So that's going to be where we start the gene. Start your gene here, okay, at the promoter. So there's a specific sequence. Binding to that promoter are certain proteins. So they'll get some proteins that come in. These proteins are called transcription factors. And the transcription factors can identify then the beginning of a particular gene. There's all different sorts of transcription factors that are unique for different genes. So a transcription factor that might be able to work on this gene, B here, doesn't recognize gene A, or C or D. It might be very specific for gene B. That's it. So if you have that transcription factor, it can help bind to the beginning, the promoter of gene B, but the others it's going to ignore. It's not going to do anything with it. Once it attaches, or there could be many of these transcription factors, and it could be a very complicated process, but the idea is that they're typically always involved in some way. An enzyme can come in, and this enzyme is going to be RNA polymerase. Now, RNA polymerase, we saw in DNA replication, it was essentially the primase. A primase is an RNA polymerase. It's going to make that little piece of RNA, but here, it's not going to just make a tiny piece, it's going to make a big piece of RNA. But it's only going to make it from one strand, and that's kind of the nice thing here is that in the other process, we needed helicase to pull them apart. We needed to keep them apart, so you need single-strand binding proteins in there. Then you needed the primase to kind of primer. Then you need the DNA polymerase to come in. You had to worry about supercoil. So all these other things were happening, right? In this case, it's good, say for you, in that it's just this enzyme. It does kind of everything. So, and it's only going to do it along one strand at a time. So what's going to happen is the DNA is pulled apart here, and we're moving, in this direction. So let's just say the promoter, we'll redraw this, okay, in a slightly different way because I'm going to show now action, like something actually happening here. So if this were the promoter over here, this bit of information, and let's redraw some arrows. Okay, so transcription factors binding to the promoter over here, the RNA polymerase will come in, and it's going to work along one of the strands. It's just going to work along the strand that would have, if this were DNA replication, have been the leading strand, and the other one that would have been the lagging strand is going to be ignored. So if you haven't already studied the DNA replication part of this, these terms don't mean as much to you, so that should come first. You should be studying that stuff first. So in orange here, what I'm looking at, this is a new piece of single-stranded RNA that's being made along a piece of DNA. So the DNA is being used as a template, and we're making a piece of RNA along it. So we're going from DNA to RNA like this. And so this RNA polymerase is going to be working along here, continuing to work, moving forward. What's going to then eventually happen is this. A piece of DNA is going to go back together again. Okay, so if this were here, it's going to kind of come back, and a new part is going to be opened up and pulled apart like this as the polymerase is working. And so what you're going to get is then this is now the piece of RNA. So after the DNA is pulled apart, it goes back together again, and the RNA detaches. So this piece of RNA was once attached here, but now it's been pulled off, and it just goes on its own. It's just temporarily bound to the DNA, and so the DNA keeps coming back together. So we don't have. That's why you don't have to worry about the supercoiling or the extra twisting of pulling the two DNA strands apart, because the whole thing doesn't get pulled apart. Only a little bit gets pulled apart, and then it goes right back together again. So you're just doing a little bit, so you don't have to worry about that. Just pull apart just enough to read this little section, and the DNA comes back together, and the new piece of RNA just kind of gets pulled off the DNA. And then in the end, when the whole process is over with, the DNA just goes back, to how it was in the beginning, and we end up with a new piece of RNA that's been made by the enzyme RNA polymerase by reading the DNA. I'll give you a few more, so that's kind of the end product. A couple more details. A little bit of space here. So if the DNA sequence, say with A, T, G, C, 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 A, G. I'm going to do that. Just a random sequence I just did here. When the piece of RNA here is made, so it's going to follow the same kind of rules. You're going to be making the new strand from the 5' to 3' direction. So you're reading the DNA 3 to 5. So all the things that were happening in DNA replication are essentially going to be the same, except it's going to be just doing it along the one strand, like this. The other difference is that when the new nucleotides come in, the way in which they pair. So in RNA, what should normally go with an A would be a DNA, we would have a T. But in RNA, there are not T's, there are U's. So this is how we would tell. This is the difference. That this is RNA, not DNA, because there's U's instead of T's. So U, A, C, G, all this would be just the same as it would. So these part would just be the same as it would be with DNA, G's with C's, and the A with the T over here. But now again, a U, because you have an A here, and so on. So this little piece here would be our piece of RNA. And we could easily tell it is from the DNA because it has the U's instead of the T's. Right, so that would be the main difference. But otherwise, everything else about it would be just the same. The sugar, if you remember back to the nucleotide structure, is a little bit different. It's a true ribose sugar as opposed to the deoxyribose sugar. But other than that, just a single strand of A's, T's, G's, and C nucleotides strung together by this enzyme called RNA polymerase. And that would be transcription, and that gives you a precursor RNA. So that gives you a type of RNA from a gene that's going to have a job. Its job is not necessarily to code for a protein. There's going to be different types of RNA. That's what we're going to look at here next. So from transcription, we'll make RNA from the DNA. Keep some of this here. What is this RNA's job going to be? Well, it could be a couple different things. Right? It's coded in the gene. So this might be a gene for messenger RNA specifically, and so it doesn't have a choice. It can't be a different type of RNA. It's going to be messenger RNA. But not every gene is for messenger RNA. This gene could be for ribosomal RNA, a different type of RNA. It does not code for polypeptides, amino acid sequences, proteins. It doesn't code for that stuff. It does something else. It's ribosomal information to make a structure called the ribosome, of the cell. That's going to be made from ribosomal RNA. It's not messenger RNA. So not all genes code for messenger RNA. They code for different things. So transcription will give us one of really three major types. There are other types as well. There are three major types we can have messenger, transfer, or ribosomal. And these are all different types of RNA made through transcription. So the process of transcription makes RNA, and the different types of RNA you may get. So who is this? It's going to be one of these. How do we know? Well, we may not know. Initially, they have to be processed first. So typically in our cells, messenger RNAs are the ones that contain the code. So there's a separate lecture, and there's one where I go into a lot more detail of decoding the genetic code. So essentially, because the idea is you're going to have this code in the messenger RNA written in the language of the nucleotides, right? Like A, U, G, C, C, A, C, A, U, G, A, C, and so on. So there's a bunch of nucleotides in a row in a sequence. So what? What does that mean? Well, it's a code. It's a code. That's what we are called the genetic code, right? So it is broken into groups of 3 little clusters. And each little cluster of 3 is a special code for one specific amino acid. All right, so that's an amino acid called methionine. So AUG codes for methionine. And so each of these, CCA, GAU, GAC, they code for different amino acids. You don't have to memorize them. In this class, usually that's more an advanced course where you would have to actually learn, all the different codes and which amino acids they code for. You'd be able to see a table, which I have in another lecture, that gives you the details of some of that and how it would be interpreted. But that's the idea. So messenger RNA contains this code. Sometimes for organisms like bacteria, all the information is coding. Yeah. For eukaryotes, organisms like us and many others, not all of it codes, and so some of it gets cut out, it gets spliced. That's also a way in which there could be more information carried in single genes, or that one gene could be used to do many things. So an old way of, there used to be saying one gene, one protein. Very, very, very old and outdated understanding of the way this works. Because what we understand now is one gene could be responsible for the code for 10 different proteins, or we could need 10 genes to really make one protein, or the gene itself may not code for a protein at all. It codes for the transfer of ribosomal RNA. So genes code for functional products, which could be messenger RNA, transfer RNA, or ribosomal RNA. The messenger RNA of those 3 contains the code for amino acids. Okay. Here. So it's a code for amino acid sequence. The sequence of amino acids. Who's first? This guy, methionine, number 1. Who's second? The one that's coded for by CCA. And then you look these up, and then you just kind of go along with the code. You don't have to, not going to do that necessarily in our course, but that's the idea of it. I'll give you a little extra exercise to do on that for additional extra credit, where you can then decode codon. So I'll give you DNA, you'll turn it into RNA, then you get the RNA code, and then you'll use one of these tables and then decode it and then come up with the amino acid sequence. You actually will do these processes. You will perform transcription and you will perform translation, and that's something that you will do as an extra credit project, because it's a little bit more involved. Okay, so just kind of keep that in mind. Right now it's just kind of definitions. So we kind of got the transcription makes the RNA, makes 2 different types. The messenger RNA contains the code for the amino acid sequence. That's one option. Another option could be that this piece of RNA that comes off here could be the transfer RNA. The transfer RNA carries amino acids and also what's called the anticodons. And that's important, right? That's a really important part. So a transfer RNA starts off with just a little piece of RNA like this. We just call it T. It's supposed to be a T. RNA. But then it gets folded, right? So transfer RNA gets folded into these little loops like this, and things called stems. So where there are two parts, actually, it actually folds back and actually joins to itself, hydrogen bonds to itself, which is what we are trying to prevent in DNA with the single strand binding proteins, for it to come back together again. Because DNA could do this, RNA could do this, and RNA does this. And that RNA folds itself into three-dimensional structures using these sort of stems and loops. And with transfer RNA, you end up with 3 loops, this, and 4 stems. Into one of these stems, which would be the 3' OH group, right? Remember we talked about the DNA having ends, and RNA also is going to have ends, a phosphate end and a OH hydroxyl end, 5', 3'. On the 3' end, the one with the OH group, that's where the amino acids get attached. So an amino acid gets attached to this transfer RNA. Down here in the bottom loop is the one called the anticodon loop. And so, say, for example, for methionine, if messenger RNA has the codon for the amino acid methionine as AUG, remember I did a little pairing up here, the complementary bases. What pairs with A? Well, we normally say T, but in DNA, but remember in RNA, it's U, right? So we'd have a U here. With U goes A, see, and then with G goes C, as always. Right? So this is the anticodon the anticodon for that codon. UAC is the anticodon for AUG. The transfer RNA has the anticodon for that particular amino acid. The codon then has the messenger RNA has the codons for the amino acids. It says what order should they be placed in. And then there's all sorts of different transfer RNAs, each one with different amino acids, and then a different anticodon code, so they can pair up. So when it says put them in this specific order, this transfer RNA comes in. And if it has the right anticodon, then good. Then it'll stay there and something will happen and a bond will form. If it's not, it says, no, you don't match. You don't pair up. Go away. Let's try a new one. And then another transfer RNA would come in and do that. That process that I'm just talking about there does not happen all on its own. It happens inside a very complex little molecular machine called a ribosome. And that's where we get to the third type here. The ribosomal RNA forms the ribosome. So the ribosome, keep this in mind, it's the only non-membranous cellular organelle. Cells have structures called organelles. The membranes form organelles, like the nucleus, mitochondria, the endoplasmic reticulum, the Golgi apparatus. All these structures within inside a eukaryotic cell that have different jobs or functions are called organelles. They're all mostly made of membranes. The ribosome is considered a cellular organelle, but it's not made of membranes, it's made of RNA. It's made of ribosomal RNA. So there's different types of ribosomal RNA, which, again, in a little more advanced class, we get into all of them, but right here, we say that there's a piece that forms what's called a small subunit. There's typically 3 other pieces that all assemble together from different genes. So like, it's a gene for one piece, it's a gene for another piece, it's a gene for a third piece. They all have to come together. And they come together with proteins, and then they form something called a large subunit. And they're not attached all the time. They're only attached when they're working and doing their job. The large subunit has components of it that are like a massive enzyme to carry out chemical reactions. And it has specific binding sites for the transfer RNA and the messenger RNA. So it's actually how all these things fit together. And so what we're kind of doing right now is we're kind of beginning the next process, the translation process. So answering the question of how do we go from RNA to polypeptide? We're translating from a language of nucleotides into a language or sequence of amino acids, right? So we're going from one sequence of information to a different sequence of information, translating that. And so that's kind of done here with these three elements. The ribosome is really the machine doing it. The transfer RNA is the part that has the decoder essentially in it. And then the messenger RNA has the actual information to say who goes in what specific order, which amino acid goes in which order. So I'm going to get to this process next, but we're going to end this particular lecture here. So we're going to still be continuing with gene expression, but it's going to be the next part of it, the translation part of it. So, which we're kind of getting into the beginnings of right now. So for transcription, remember, it's just the process of DNA coding for information, and that information being copied into RNA. An entire chromosome is not copied, only sections of the DNA called genes. The beginning of each gene is a region called a promoter. It's identified by proteins called transcription factors, who identify the beginning of each gene, where they start, and whether they'll be read or not. And then we have RNA polymerase who comes in, and it pretty much carries out all the rest of the work, to pull the strands apart, to read the DNA, to make the RNA, and then terminate the process. When a piece of RNA is made, it's a precursor piece. It's set to be messenger or transfer or ribosomal based on its code, but it's not really functional until something happens to it. So the messenger RNA sometimes has pieces cut out of it. It has extra things added to it to become functional. The transfer RNA has to be folded and then have amino acids attached to it to become functional. And the ribosomal RNA also has to be folded like this, far more complicated, folding, and then proteins bind to it as well, and then it forms these two little molecular machine pieces, right, that are called ribosomes. And that's kind of where we'll begin the next part on translation.